Hi, welcome to the Genes and Tissue Culture Technology. So this is our first online session. I will introduce you to the general overview of the module. So this module is basically studying the behavior of animal cells. So we'll look into specifically mammalian cells, but not plant cells, on the tissue culture techniques, as well as the platform for genetic and protein engineering. Apart from that, we will also look into some of the molecular tools, for example, bioinformatics for DNA and protein sequence analysis. So there are altogether 4 topics for this module. We have principles of cell culture, number 1. Number 2, gene technologies, followed by protein technologies, and the last topic would be on bioinformatics. So first of all, I would like you to introduce yourself on the Padlet wall in the Times course page. So I have seen a few of you already introduce yourself, including Nigai, Ivy, Jeff or Jeff. I'm not sure who is this, Gary, Vivi, and those from Drug Discovery. Okay, it's very important for me to know all of you and for all of you to know your classmates. This is a module shared by semester 4 and 5 students, so I hope everyone can introduce yourself on this Padlet wall. So, what is so important about cells? For our Genes and Tissue Culture Technology module, it is an extension from the Eukaryotic Cell Biology module. So we'll look into the in vitro propagation of cells. Okay, so there are many types of cells in the human body. For example, fat cells, brain cells, red blood cells, or nerve cells. So we'll also study about other cell types like cancer cells, hybridomas, stem cells, or transfected cells. And according to our title of our module, Genes and Tissue Culture Technology, we'll also look into the genetic material, for example, the genes. Okay, so let's say the Human Genome Project, which is a sequenced human genome. So what can we derive, or what are the applications from this Human Genome Project? So there are many applications in this field. It helps us to understand the disease, for example, from the human genome, to identify mutations linked to different diseases, to identify oncogenes, or to select drug targets to predict their effects, or we can use it for personalized medicine. So more importantly, we will be using genetic engineering as a tool to manipulate cultured cells. For example, I have given 2 samples here. So these are very new technologies in which they can reprogram somatic cells to become induced pluripotent stem cells. So what are induced pluripotent stem cells? They behave very similarly to the embryonic stem cells, but they are derived from the adult cells. So you can see that they can use different genetic technologies to reprogram a terminally differentiated cells, for example, human or mouse fibroblasts, into stem cells. So we'll look into this stem cells application in chapter 2. So tissue culture is a very important technique in which they will teach you how to grow cells, tissues, and organs in the lab. So how do we obtain these cells? So usually we can isolate cells from pieces of tissue, or we can isolate them from patient samples like peripheral blood. So for example, from the blood, we can isolate B cells, T cells, dendritic cells, macrophages, or natural killer cells. And then we can induce them to grow in the lab using optimal conditions, for example, providing them with the suitable medium, pH, temperature, growth medium. So we can culture these cells and understand their behavior in the lab. So tissue culture is a general term we use to include organ culture as well as cell culture. So in order to culture cells in the lab, we need to have conducive environment. So we'll culture the cells in glass or plastic culture dishes, and usually in the form of on the support of the solid medium or the liquid medium to provide the essential nutrients for the survival and growth.